very interesting scientifically to think about. And when they do that, the immune system, which actually is hidden, it normally does not get into your brain. It's a quiet area. It's so complicated, even your immune system cells don't bother going in there for the most part. So the immune cells in the body, all of a sudden at age 50 in this woman, are able to see a brain protein that they think is a virus. They think there's a cell that's got something wrong inside it, and it killed her tumor. And part of that killing had to do with recognizing this protein as a foreign, abnormal protein. It really wasn't foreign. Okay, it was really made inside the brain, but since the immune system doesn't go in the brain normally, it could see this as if they had the flu inside a breast cancer cell, right? And in fact, we know by a lot of research now, in the Rockefeller Hospital over the last 15 years or so, that this attack is very specific and very potent. So these patients are like this rare Rosetta Stone, where they're actually recognizing their cancer and getting rid of it. Now, the reason we know that's going on is because we think in 1 out of 100 of these patients, that immune response somehow does break the blood-brain barrier, get into the brain where it shouldn't be, and thinks it found more tumor cells. But in fact, what it's actually seeing inside the brain are the cells, the neurons inside the brain, that were normally making this protein that the cancer cells stole. And when it sees those neurons, it starts to kill the patient's own neurons. And in this case, they're neurons that control how you move. So it's a part of the brain called the cerebellum, which controls your motor movements. So these patients, and we see them, many of them in the Rockefeller Hospital, even though they're esoteric patients, they have the potential to teach us about this hidden iceberg question of tumor immunity, and also the potential to teach us about how the immune system can get into the brain and cause autoimmune disease. And there are many autoimmune diseases we would like to understand better, like multiple sclerosis is one you might have heard of. So I'll just show you some other pictures of how efficient this tumor immunity can be. This is a CAT scan of the chest here. It's like a cut through somebody's chest. This is the chest wall, and this black thing here is the lung. And it's supposed to be black because it's got air in it. But there's a dot here. So this was a patient who smoked cigarettes. I'm proselytizing, not smoke here. All right? This patient smoked cigarettes, got a nasty tumor that smokers, only smokers get, called small cell lung cancer. It's widely metastatic when it's first diagnosed, and most people kills them within 2 years. In this patient, it was picked up on a routine CAT scan because the patient was a smoker. The patient was sent home for treatment, and when they got sent home, they developed one of these paraneoplastic syndromes. And they came back for treatment, and the cancer was disappearing. And at a second point later, they came back and the cancer was gone. This patient never received any treatment at all. So this is an example where we can catch it, now that we understand a little bit more about the autoimmune disease and the paraneoplastic syndrome. We can actually catch this cancer in the act of disappearing. Okay, this is the immune response showing, you can make a brown stain out of the immune response and see that there are antibodies in these patients recognizing the tumor, whereas most patients who have cancer don't have antibodies and don't recognize their own tumor. So, a little more detail here. One can look in the patient's blood and actually have a way of diagnosing this paraneoplastic syndrome because the antibodies that circulate in the blood are part of this immune response. And those antibodies can bind to specific proteins. And not only do they bind the protein in the patient, but you can grind up tissue from a tumor cell line, run them out in a gel like this, and show that these patients, but not normal people, the first one is me. I have no antibodies against tumor proteins, but these different patients do. You can see different size proteins here indicate that different patients are recognizing different antigens, different proteins inside the tumor. So this experiment, and I'll maybe come back to this a little bit at the end, allowed us to use this methodology, and I'll explain this a little bit more later, to do modern molecular biology on these syndromes, okay? Because by using antibodies to see the proteins, we can also use the same techniques to clone the genes that are encoding this protein that's made inside the patient's tumor and normally made inside the patient's head. And once you have the gene in hand, you can start to do all kinds of modern science, 21st century science, to understand what's the nature of the diseases. So this one turns out to be an antigen associated with breast cancer. This is the antigen associated with lung cancer. And using this sort of black and white technique of the antibody and the immune response to identify for your weapon proteins, you can get the genes for each one and ask, what are they? So there's two different things you can do with these genes, all right? One is to use them as tools to make better understanding of the nature of the disease, to try and come back to the bench, to try and treat cancer. And the other is to understand the nature of the genes, to understand what is going on in these syndromes. Why would a cancer cell express a brain protein, especially if it puts the cancer cell at risk for being seen by the immune response and eradicated? Okay, so our laboratory is interested in both of those questions. How could we use these genes to make better treatments for cancer? And what is going on with these genes in terms of the science and what does it tell us about the cancer and what does it tell us about the brain? What's special about these genes? Okay, so I could go on for hours, but I'm not going to do it. I'm going to mostly tell you about how we've used these genes to try and develop new treatments. To go, this is the bedside, this is the bench. Having gone to the bench and cloned the genes, we're now interested in seeing if we could use them to understand how this hidden phenomenon of tumor immunity, where there's no clinical hint of its existence except by this Rosetta Stone of these esoteric paraneoplastic syndromes, how can we use it to capitalize on that for the general population of cancer patients, okay? So to explain that, I have to teach you a little bit about how the immune system works, okay? So the immune system has antibodies in it that circulate in your blood, and it has cells, special cells called white blood cells, right? You know in your blood you have red blood cells that carry oxygen. You also have white blood cells, and those are very special cells that have a kind of an intelligence to them. One of the intelligent things they do is they're able to do this process that I mentioned before. So they're able to see what's going on inside of the cell. So this cell has abnormal proteins like a brain antigen that it's not supposed to make, or an abnormal protein like a virus antigen. These cells can see it. How do they do that? Well, they have a very special receptor on the surface of these cells, and that receptor is able to see what's inside of the cell, because what's inside the cell is proteins, and proteins are always chopped up and turned over. And when they're chopped up and turned over, little pieces of it get captured, like this blue piece, and put on the cell surface, on a receptor, on every one of your cells has receptors on it that display what's going on inside it. Why do they display what's going on inside it? Just for the immune system to be able to see. If there's something abnormal going on, these cells can see it. So their receptor puts little fragments of all of your cell proteins on the surface. And if you get infected with a virus, the virus protein will get put on the cell surface. And this T cell has a chance to see it. Now this really works, okay? This is a movie here. This is a cancer cell called a melanoma. It's a nasty cancer cell. And here's one of these killer T cells. Those are called a cytotoxic T cell, or CTL. And this movie shows you something interesting here. So just watch here as these T cells, there's two of them, they home in on the cancer cell. And they recognize via these receptors what's going on inside the cell. You can't see that because that's at the level of the molecule. And bingo, these like balloons pop out of the cell. Okay? This is the cell dying. And in fact, they die via a special kind of death. These cells induce the cell, the cancer cell, to commit suicide. And it's called apoptotic death. And it's a very active death that happens. It's a special kind of death, and I'll talk to you a little bit about that. So how does this cell suicide work? And how do these T cells work? Okay? So this is a fun, complicated slide, but the bottom line is here's the T cells. They can't just go out and kill a cancer cell on their own. They're so destructive that they're actually dangerous. And one of the dangers is they might start recognizing their own cells because of autoimmune disease. So they're held in very careful check by your body. And they're held in careful check in the lymph nodes. Have you ever gotten a virus and felt like under your neck, the nodes swell up, right? Those lymph nodes, we've all felt that. They hurt sometimes. If you get a really bad sore throat, like a strep throat, you know, it swells under here, you can feel it, it's tender. Those are your lymph nodes. And inside the lymph node, there's millions of cells, and that's a center for control of these